so this is what this looks like. The first step in the breakdown of these molecules is the hydrolysis of these ester linkages. So if we bring in here 3 molecules of water, we can then generate the glycerol by itself. And we're going to generate the 3 different fatty acids. R1, R2, and R3. So we've got 3 of these separated fatty acids, and the metabolism of these is then going to continue. Now, this, of course, is a difficult bond to break. The ester bond can be broken chemically, but it takes extremes of pH and temperature. And so this reaction is catalyzed by what are called lipases. And these lipase molecules are actually specific for one or the other of these ester bonds. So there's different lipases that carry out this hydrolysis reaction at the different positions on the glycerol molecule. So that gives rise to the 3 fatty acids within the muscle cells of our body. Then the next step here is going to be to activate this fatty acid. It's not the bare fatty acid that's metabolized. It is an activated fatty acid. And the activation process looks like this. We're going to start out with the fatty acid of this structure here. And we react this with ATP using an enzyme. And we release pyrophosphate, the last 2 phosphates on the ATP molecule. This then gives us the AMP intermediate, where the energy of the ATP now resides in this bond between the carbon and the AMP molecule. And then in a second step, we are going to bring in coenzyme A with its sulfhydryl group, as you can see here, and that's going to form the acyl coenzyme A molecule, which looks like this. So there is the acyl coenzyme A molecule, and this is the activated form of the fatty acid. You remember that in glycolysis, we needed to prime glucose to glucose 6-phosphate to initiate the glycolysis pathway. So we had to use ATP to get back a larger amount of ATP later in glycolysis. In the same way, we're needing to prime the fatty acid molecule to carry out the subsequent reactions. And here, it's a similar principle. We are using the energy of ATP to carry out this priming reaction. Now, this is a little bit different. We talked about this way of using ATP back in the early chapter on metabolism. But instead of breaking off the terminal phosphate and generating ADP, I should indicate the other product here is AMP. Instead of doing that, we chopped off the 2 terminal phosphates to make pyrophosphate. This, using another enzyme, is hydrolyzed into 2 inorganic phosphates with water. And then we use the energy in this bond to form the bond with the coenzyme A molecule. So we are using the energy of ATP twice here. In metabolic accounting, we would count this as 2 ATPs, or 2 high energy bonds. So remember that. Whenever we use the energy of ATP in this fashion here, that is going to be equivalent to using 2 molecules of ATP or 2 molecules of high energy bonds.
Where's the second molecule of ATP?
Well, there's not 2 physical ones, but it is equivalent to 2 because we use it twice. we break it up first into pyrophosphate, then we hydrolyze that. That's an exergonic reaction, so that pulls this reaction forward by the law of mass action. Then that's a high energy bond, and that is used to form the coenzyme A. If we want to regenerate ATP from AMP and these two molecules of phosphate, we have to get two ATPs into the mixture. If you look back on those reactions in the chapter, I think it was chapter 15, Introduction to Metabolism, you have that business. So whenever you use the ATP energy in this fashion, it counts twice as much as if you just go to ADP in one phosphate. So keep track of that. By the way, I'm sort of going along here with the pictures in the book. So a lot of this is in your textbook, so I'll just sort of follow it as I'm doing here, but I'm going to write a lot of this on the blackboard as well today. Okay, so this is the activation process, and we are now ready to begin the actual metabolism. But this particular step here has occurred in the cytoplasm. So the cytoplasm of the muscle cell. And now the further reactions of the metabolism of that fatty acid coenzyme A now occur within the mitochondria. So we have to get this molecule into the mitochondria, and there is a specific molecule that does that, that carries out the transport into the mitochondria. And I think what I'll do here is just show the slide of that. And here's the carnitine molecule. This again is from your textbook and is on the blackboard. So here we start out with the acyl coenzyme A molecule. Here is the carnitine, and we simply transfer the acyl molecule, the fatty acid, to the carnitine as an intermediate and release the coenzyme A in the cytoplasm. Now the next step is the transport inside of the mitochondria, and this summarizes all of that. You start out transferring the fatty acid acyl group from the coenzyme A to the carnitine, as we've just shown. That then is transported to the inside of the mitochondria by a specific protein called the fatty acyl translocase, and it brings the carnitine in here. Then it's transferred back to coenzyme A, and carnitine goes back out again. So it's a nice little cycle there, and the net result is we get the fatty acid inside. The fatty acid coenzyme A, as a matter of fact, inside, but we don't actually transfer the coenzyme A, we pick up a new one on the inside. So the net result is we've now got our fatty acid on the inside of the mitochondria, where we can begin the actual process of metabolism. Okay, so let's take a look at this, and you can follow along on the board, but it's also written up here in your textbook. So let's go and look at what this whole reaction looks like here. So I'm going to draw a molecule of a fatty acid in somewhat abbreviated form here. So here is our coenzyme A. Here is our thioester linkage. I'm going to draw 3 methylene groups, and the rest I'm going to leave as an R group. Now, if this had 16 carbons, this would be palmitic acid. That's the name of the saturated fatty acid with 16 carbons. So imagine that this is palmitic acid. This would be the first 4 carbons. We then need to have 12 more carbons in the rest of the chain I'm abbreviating as R to make the full molecule. You'll see why I chose 4 in just a second here. We then begin the first step, and I'd like you to kind of focus on the logic of this reaction. We begin first with an oxidation reaction, And we need an oxidizing coenzyme to do that. And so I'm going to use an enzyme that has an oxidizing agent, FAD. And we've seen FAD used before in the mitochondrial respiratory chain. This then forms an FADH2. This then generates this molecule here. And so we have a double bond that's formed as a result of this oxidation. And here is our R group down here. Better make sure I've still got my 4 carbons that I'm showing. But we've oxidized this single bond right here into a double bond in that fatty acid. So that's the first oxidation. Then the second reaction is simply the addition of a molecule of water. And that is going to form this structure here. So we're adding water to this double bond here. Here is the alcohol that we get there. And we still have our 4 carbons showing, and then the rest of them in the R group. So this now, by the way, should be a CH2 at that position. I think that's all right now. So the next step here is going to be to carry out another oxidation. So this time we're going to bring in a NAD plus group, generate an NADH, and the result of this is going to be a ketone. Okay? So, the final step now is going to be to split this molecule into 2 pieces, and we'll be ready to continue. So what we're going to do now is, I'll bring a long arrow here. We're going to bring in another molecule of coenzyme A with its sulfhydryl group. The top part of this is going to be an acetyl coenzyme A. So this part right here, we're going to be breaking the bond right there. And then the bottom part is going to look like this. Okay, so there we go. We've got these 2 different pieces now. We've got one molecule of acetyl coenzyme A with 2 carbons. And we have a new fatty acid with 2 carbons fewer than the original molecule. And let me just indicate here, if this was C16, palmitic acid, this molecule here would be a fatty acid with 14 carbons. We just call that C14, okay? So we've just shortened our fatty acid by 2 units, the equivalent of the acetyl group. Now, we bring this all the way back. This becomes the new fatty acid that now has 14, and we continue that cycle one more time, and then we continue it until we've broken down the fatty acid all the way into acetyl coenzyme A molecules. So let me ask you this. For C16, if we chop it, how many acetyl molecules are we going to get? Well, we'll get half of that because each of those carries 2 carbons, so that would be 8. So how many of these cycles do we have to go through to completely break this down into the acetyl coenzyme A molecules? We had 16 carbons.
8.
We break it down into 8 pieces of acetyl-CoA. How many cycles?
7.
7, exactly. So the number of cycles is 1 less than the half of the number of carbons. Now, we'll see later, the reason that most of our fatty acids have an even number of carbons is that they are synthesized from acetyl coenzyme A, and then when they're broken down, they are broken down into acetyl-CoA. And so since acetyl-CoA has 2 carbons, most of the fatty acids have an even number of carbons. Okay, so that completes the reaction. Now what I want to do is to go ahead and make a complete summary of this, and I'll do this for the C16 that we're talking about here. So we're going to start out here with palmitoyl, coenzyme A. If we go through 7 cycles, as I've indicated there, we are going to use 7 FAD molecules. We are going to use 7 NAD molecules. We are going to add 7 molecules of coenzyme A, and we're going to add 7 molecules of water. And this is all going to go to 8 molecules of acetyl coenzyme A. Plus 7 FAD H2 plus 7 NADH plus a proton. I don't know how many protons. I'll leave an abbreviation there. So that is the summary reaction for the metabolism, the complete oxidation of the C16 fatty acid, palmitic acid. Just let's focus on the logic of this one more time. We take the saturated fatty acid, we oxidize the single bond to a double bond, we add water to form the alcohol, we oxidize the alcohol to a ketone, then we chop it into acetyl-CoA and another thioester, coenzyme A ester, that repeats the process. So what we get out of this whole process is 8 molecules of acetyl coenzyme A, 7 FADH2s, 7 NADHs. Now, I'm big on metabolic accounting. The bottom line with any metabolism is how much ATP do you get out of here? And fatty acids, above all other molecules, are in the business of producing energy for ATP synthesis. So we really want to do a metabolic accounting here. This is the first stage in that metabolic accounting. And what I want to do next, if I can find this, is I want to do some summary things. What's going to happen to all the stuff that we've produced here? What's going to happen to the acetyl coenzyme A, first of all? Where's that going to go? Citric acid cycle. What's this stuff going to do? We're in the mitochondria now. That's the electron transport chain. And likewise, NADH is the electron transport chain. Let's first of all go back to the citric acid cycle and give a summary of that citric acid cycle for the acetyl-CoA. So I gave that before once, but let me put it down here. So for every molecule of acetyl-CoA, if we enter the citric acid cycle, we are going to react this with 3 molecules of NAD plus 1 molecule of FAD plus 1 molecule of GDP plus 1 molecule of phosphate plus 2 molecules of water. And that is going to go to 2 carbon dioxides and 1 molecule of coenzyme A SH and 3 molecules of NADH and 1 molecule of FADH2 and 1 molecule of GTP. And I'll just put, I think we have some protons over there. I'm not particularly interested in the big picture here of how many protons we have. Now, let's account for this. We've got our balance sheet for acetyl coenzyme A metabolism through the citric acid cycle. This is the balanced equation for the citric acid cycle, right? And you should have this on your fact sheets, as a matter of fact. I'd like you to have a lot of balances. We had another balance using pyruvate into the citric acid cycle. That's identical to this, except 1 more NAD is produced. For the acetyl-CoA, this is the balance sheet. Now, what is the ATP yield of this? How many ATPs do we get from each NADH? 2.5, right? And so this is going to give us 7.5 ATP molecules. I'll put 2.5 here. That's per NADH. How many do we get from the FADH2? Remember that one?
1.5.
That's 1.5. So this would be 1.5 ATP. And and then this GTP, we count that as equivalent to an ATP. That can be interchanged with ATP. So that's 1 more ATP. You put all these guys together, and the net yield is going to be 10 ATP molecules. Okay? So that's going to be our net yield. Now, let's go back over here, and we'll finish this off here. If we get 10 molecules of ATP from each acetyl-CoA, I think you can see we are going to get 80 ATP here. These 7 FADH2s, that will be multiplied by 1.5, and so that's going to give us a total of 10.5 ATP. And then we're going to get 2.5 for each of these. That's going to give us a total of 17.5 ATP. Now, let's add all of this together, and I guess I don't know anything to do but get rid of that little thing for the time being. And if we put all this together, I'm going to give you a final balanced equation there. So this will be palmitoyl, coenzyme A, plus 23 molecules of oxygen, plus 108 ADP, plus 108 phosphate. And this is going to generate 16 molecules of carbon dioxide, 108 ATP, plus a coenzyme A molecule, plus some number of waters that I haven't worked out. Okay? Now, the bottom line here is that fatty acids are enormously energy rich. 1 molecule of palmitic acid is able to generate 108 molecules of ATP. So that's the bottom line from a metabolic accounting standpoint. We generate an enormous amount of ATP. And if you look at the old exams, I've given a number of questions there that use different fatty acids than the palmitic acid and ask you to do this type of metabolic accounting. Yeah?
Where do the 23 oxygen molecules?
Okay, that's a good question. So that, what we do here is that each of the FADH2s use a half of an oxygen molecule. Remember, the FAD is a 2-electron carrier. Oxygen is a 4-electron reactant. So we add up all of the NADHs and FADH2s, and we divide by 2. Now, this would be here 14 of these 2, and so that would be a total of 7 O2s there. But then, and I'm sorry, but, oh man, got stuff all over the place here. When we look at all of these guys here, how many oxygens is this going to be? That's going to be 3 of those and 1 more of that. That's 4. This would be 2 oxygens here. Okay. We multiply that 8 by 8, so 2 times 8. This is 16 oxygens, and 16 and 7 is equal to 23 oxygens. So the bottom line is you add up all of the NADHs and FADH2s, and you divide by 2, because each one of them reacts with half of a molecule of oxygen in the mitochondrial respiratory chain. Okay? Is that all right? So that's our bottom line right there. We've got 108 of this. Remember that if we started out with just bare palmitic acid, we would have to subtract 2 of these because of the priming reaction. Because of this priming reaction that we did up here, if we write a balanced equation starting with palmitic acid rather than palmitoyl coenzyme A, the answer would be 106 ATPs, because this priming reaction up here is going to, whoops, that's not the priming reaction. That's the entire thing right there. Here is the priming reaction here. That's going to take the equivalent of 2 ATP molecules. Okay, let's see where we are here. I've just put a number of slides from your textbook in here. That just names all of the enzymes that we put on the board over here, showing their reactions. Okay, we're ready for the next step in fatty acid metabolism. Any questions on this before I go on? Okay, so the next step is to talk about a metabolic condition called ketosis. Ketosis is a very serious condition that results when we are starved for carbohydrates. You can get, a diabetic patient can get this when there's not adequate insulin that allows uptake of glucose into the cell. That would starve the cell for glucose and other carbohydrates. You can get this during starvation. If you eat an extreme Atkins diet, eating nothing but fats, you will get ketosis. This can be diagnosed by doctors by the smell of acetone on the breath of the patient. And so that's a typical test, or early test, for the condition of ketosis. What happens during ketosis is, because there aren't any carbohydrates, the citric acid cycle becomes depleted of its intermediates. And remember, to make the citric acid cycle intermediates, we don't need to make them if the cycle is already filled with intermediates. They regenerate. But if it gets depleted, we make it through the anaplerotic reaction from pyruvate to oxaloacetate. If we don't have any carbohydrates or any glucogenic amino acids, we're not able to do that. We deplete the citric acid cycle. That shuts down. And that initiates the metabolic condition called ketosis. So now what we're going to do is to look at this process of ketosis here. And switch pens here. So I'm going to start here with 2 molecules of acetyl coenzyme A. remember that we can make lots of acetyl-CoA by metabolism of the fatty acids. If it can't enter the citric acid cycle, we don't get all that beautiful stuff. We don't get that 108 ATP molecules. Instead, the following happens. We combine initially 2 molecules of acetyl coenzyme A, and we generate this molecule here, which is called acetyl, or it's acetoacetyl coenzyme A. So that's simply 2 molecules of acetyl combined together, and we release one of the coenzyme A molecules. We then go ahead and combine this with another molecule of acetyl coenzyme A. And we make this molecule here. So this has got S coenzyme A up here, double bond there, CH2, C, OH, CH3 there, CH2, COO minus, and we release another molecule of coenzyme A, okay? And then we continue along here, and now we release another molecule of acetyl coenzyme A, and we generate this molecule here. This is called acetoacetate. Okay, almost done here. And then there are 2 possibilities for what happens to the acetoacetate. One of these possibilities is that we release carbon dioxide and we form acetone. So that's going to be CH3, C double bond O, CH3. So that is what you can smell on the patient, on the breath of a patient that has ketosis. The other possibility here is that we can go ahead now and reduce this molecule with another molecule of NADH, forming NAD+, and this is going to generate, and I'll put that in the same way that it did before, CH3, COH, and COO minus. That's the bottom part of this molecule. And so let me give some names here. I won't put all of these down. This is acetoacetate. This is D3-hydroxybutyrate. And this is acetone. Now, all 3 of these molecules are called ketone bodies. So we just call these collectively ketone bodies. I don't know why they call it bodies, but that's just a historical name for them. And so these are what are produced when we're in a state of ketosis. Our citric acid cycle is completely shut down, and this is all we got. Now, you can see that the metabolic consequences of this for the production of energy are going to be rather extreme. We still are going to get these ATP molecules from, if we started out with palmitoyl and completely metabolize that during a stage of ketosis, we would still get this reaction to occur. This reaction to this point does not require the citric acid cycle. We would still get the 10.5 ATPs from the FADH2, because our electron transfer chain is still working, we'd still get these 17.5 ATPs, but we wouldn't get any of those 80 ATPs because all of this acetyl-CoA would be converted into the ketone bodies, which are on the slide that's covered up right now. Okay? So we could continue to undergo the metabolism of fatty acids in a state of ketosis, but instead of getting 108 ATPs, we get it whatever the sum of this is, which I guess is 18 ATPs. So we get many fewer ATP molecules. The other thing about this is that these molecules, these ketone bodies, the acetone, the hydroxybutyrate, the acetoacetate, they can themselves be used as an energy source by certain tissues of the body. Under some circumstances, these ketone bodies can be transmitted in the bloodstream to the heart, and under extreme circumstances, that can use these ketone bodies as a source of energy, so that is possible. Let me just look at a few. The same thing that I've just drawn is on this slide up there, as you can see. And this just shows the pathway that if we want to break down these ketone bodies, sending them to a specific tissue, then I think you can see that we can take the hydroxybutyrate, convert that to acetoacetate, acetyl-CoA, and then make acetyl-CoA. Now, if the tissue we're sending it to still has a a citric acid cycle, we can then use that. And our heart will preserve its citric acid cycle under all possibilities. So if one tissue is in a condition of ketosis, it might send the ketone bodies to another tissue which is not in an extreme case of ketosis. Yeah?
Is the acetoacetate also considered a ketone body or just acetone?
I think I would consider it a ketone body. That's kind of a semantic question, but they're all interchangeable, as you can see here, both in their synthesis and their breakdown. Okay? Okay. There's one other thing I had in this part of the talk. Yeah, this is, by the way, as I said with the amino acid metabolism chapters, we're doing kind of a stripped down version of these chapters. I'm not covering everything in the chapter. I put out a new syllabus yesterday that gives the specific page numbers that you're responsible for in the chapters. And that is precisely the material I'm covering in lecture. So for all practical purposes, what I've covered in lecture is what you're responsible for in the next exam. And you can supplement that, and you should supplement that with the indicated pages in your textbook. This is a picture that I found pretty fascinating. This just shows, for a person of 154 pounds, where all of the energy is located in the various organs of the body, broken down into carbohydrates, glucose or glycogen, fats, and proteins, mobilizable proteins, they call them here. And as you can see, there's some energy in glycogen, but this is in kilojoules here. In the parentheses are kilocalories, but I think you can see there's some energy in glycogen. But look at the fats. That's the big deal. So a normal person is carrying around that much kilojoules of energy in the fat tissue compared to all this stuff in the other forms. There is a fair amount in the proteins, but not nearly as much as what you see in the fats. And of course, during a period of starvation, we first use the fats and metabolize them, and then at some point you have to go and begin burning the protein in your muscles in order to survive. So it's a combination of living off the fat that you've stored and the protein that you have in your muscles that would get you through a period of starvation. Okay, I think that's a good place to stop then. I'm going to take up next the synthesis of the fatty acids. Yeah.
When you were talking about ketosis, that's gonna be only with carbohydrates, right? In the absence of fatty acids.
I'm sorry, I didn't hear you.
So ketosis will be only driven by carbohydrates. So we're not using any fatty acids in there.
No, when you're in a state of ketosis, you have no carbohydrates.
Oh, okay.
So ketosis is the absence of carbohydrates, which shuts down the citric acid cycle, and everything flows into these ketone bodies